diversity and the sort of importance of the diversity for the performance of these ecosystem functions have made understanding how this diversity comes about one of the really central challenges in quantitative community ecology. So we'd like to understand the diversity sort of for its own sake, but also because the diversity is key to these important processes. And so the traditional approach to explaining species coexistence assumes that species interact in a pairwise fashion. And I'll try to diagram out for you that traditional workflow in this slide. So an ecologist will go out to the field or to their test tube and measure all the pairwise effects of every species on every other. So that's what I'm drawing here. These circles are different species, and these red arrows are different ecological interactions between the species, whether they be competition or facilitation, etc. So this is, of course, like a directed network. And then you can plug these interactions into some ecological model, oftentimes the generalized Lacovol-Terra model, but there are many alternative choices, and then use that model to predict community composition and hence coexistence. So here what I mean by coexistence is just the species that I'll call coexisting are the ones that have some positive or some non infinitesimally small abundance at the end of a simulation of these differential equations, say. And so, unfortunately, this traditional approach often fails and instead predicts widespread exclusions because of strong interspecific pairwise interactions between the species. So, there's been a long tradition of work using annual plants in the field, sometimes in Southern California, but there's now many different geographic areas where these experiments have been done. And the basic observation is that the pairwise interactions between different species are too strong to predict coexistence, even though the experiments are done with sets of 5 or 10 species that we know coexist in those same fields, in the same grasslands. And so that's a little puzzle, right? What is it that we're missing about these species that is producing coexistence that we're not capturing in our experiments? There are now some similar observations from microbes in the lab that are from more recent experiments. And here the observation is that, talking about one specific nice recent paper about this, in a community of 12 coexisting strains that we know coexist in these exact sort of test tubes where we do the experiments, the majority of pairwise competitions don't exhibit coexistence, right? So one of the members of the pair outcompetes the other. And so there's some process that's happening in the diverse communities that's not necessarily happening in the pairwise communities. And in fact, these kinds of dynamics, this relationship between strong interspecific pairwise interactions and the lack of coexistence, has been predicted by theory, specifically, started by Robert May in 1972, essentially showing that as communities become more diverse and the interactions become stronger and more variable between them, you're very unlikely to see stable coexistence of the whole community. So what explains this mismatch? What explains the mismatch between predictions based on pairwise experiments or pairwise interactions and diverse coexistence? Of course, there's many possible explanations, and in ecology, the right answer is often it depends on the exact system or the exact circumstances. But I'll briefly describe two that could be going on, especially in the microbial settings where we sort of have a bit of a better handle on what's going on. So one is the idea of an interaction chain, and then the other is the idea of a higher order interaction, which I don't think will be a big surprise to this audience. And it's also going to be the focus of the talk, given the title. Okay, so what explains this mismatch? The first possibility is that there's some pattern in the in the pairwise effects between all the species that produces coexistence at the whole community level. So the sort of prototypical example of this kind of interaction pattern is the rock, paper, scissors game, where each species, whether in the rock, paper, scissors game, can unambiguously beat the next one. But when you grow all three together, none of them can grow enough, can outcompete all the others, gain enough of an advantage to outcompete all the others because they're being beaten in a cycle. And of course, there's more general versions of this applying to many, more species than three, etc. So this is one possibility. There's some sort of connected interdependence amongst all the pairwise interactions that produce coexistence when you grow all the species in the same test tube, say. The other possibility is that the strength of the pairwise interactions is just fundamentally different when you have a third or fourth species around. So that's the idea of a higher order interaction. And importantly, this doesn't rely on any time scale. There's really an instantaneous change in, say, the interaction between these two species when this third species, where the blue arrow is originating from, is in the system. So I think that's probably familiar to this audience, so I'll keep stepping right along. But the idea is just that, on the left-hand side, these interactions are fundamentally pairwise, and so they could, at least in principle, be measured from the pairwise experiments. But on the right-hand side, they can't be measured from two-species experiments. And so as a result, because they can't be measured from two-species experiments, they're just fundamentally missing from this traditional approach. But I would say, the idea of higher order interactions in ecology has been around for 50, 60 years or so, and they're often argued to benefit coexistence. And so one nice experimental setting in which it's sort of clear they do, to give you an idea of how they might arise in ecological systems, is antibiotic competition between three different microbial strains. So in this setting, one strain produces an antibiotic that kills a sensitive strain, but a third strain secretes another compound into the system that degrades the antibiotic, protecting itself, but also protecting the sensitive strain. Right? So that's a very clean example of a higher order interaction that you may not have expected if you ran all pairwise experiments. But it's not obvious that all higher order interactions in ecology need to be like that. So returning to our annual plant system, you might expect there to be some competitive effects from this green plant on this gold plant, but that when this green plant is also competing with this purple plant, its phenotype or its character changes in some way. Say it grows, further, it grows taller because it's experiencing competition for light. And that could potentially amplify its competitive effect on this gold plant. And in that setting, it's less clear exactly what these higher order interactions are going to do for coexistence. So that's the first big knowledge gap I want to try to address in today's talk, is that, even though they've been studied for a long time, we lacked, before I started working on these sorts of questions, rigorous theoretical expectations for how these interactions shape species coexistence. We also have limited evidence for how and whether they operate in plant communities, even though this is one of the systems where we know the most about coexistence and species interaction. So these are the big two knowledge gaps I want to try to address. And specifically, I want to address three questions. First, just how do higher order interactions affect coexistence in more generic ecological models? Second, what kinds of higher order interactions actually successfully stabilize diversity? And then third, do higher order interactions structure the dynamics of annual plant communities? So I already told you a little bit about this microbial experiment, and there's been some associated theory motivated by this experiment that shows that these kinds of higher order interactions are good for coexistence. They're likely to promote the coexistence of all three strains, but it's less clear how more generic mechanisms like the one I just told you about, phenotypic plasticity in response to competition, might impact coexistence. And so that's the first basic question I want to try to address. How do unstructured higher order interactions affect species coexistence in more flexible ecological models, and models that are not tailored to the underlying microbial biology of this producer-sensitive degrader example? So to address this question, we can consider a generalization of the Bloch-Terra model with higher order interactions or higher order terms. So the basic biological processes, so this is a big system of differential equations, and there's three basic biological processes that go on. There's some intrinsic growth rate that species I grows at in isolation, so how fast does it grow exponentially when no one else is in the system. There's all the pairwise interactions in the system, which are controlled by these Aij coefficients. So it just tells you how species J affects species I. Can be competitive if the Aij are positive, or facilitative if they're negative. And then there's some special pairwise interactions in the system, specifically when I equals J, which measure the carrying capacities or the level of intra-specific competition in the system. And then last, I model higher order interactions in just the simplest possible way by multiplying together triplets of species abundances so that the coefficient Bijk measures the joint effect of species J and species K on species I. Or alternatively, you can think about Bijk as being how species K changes the effect of species J on species I. So throughout the talk, I'm just going to set the self-regulation parameters to 1, these AII parameters, just to set it an intrinsic scale for the abundances. And that means exclusions, species that are being sent to 0 or very small density, are going to come about from strong interspecific interactions. And to choose those parameters, I'm going to take what's now a classic approach in this field, motivated by this Robert May work, which is to sample them from probability distributions with specified means and variances. And the idea is then to derive the coexistence properties of some equilibrium state from the statistics of that distribution of interactions. So here are two possible examples of what the interactions might look like. One where the mean interaction strength is relatively small, so competition is relatively weak in this system, but also the variation in interaction strengths is also relatively small. So in some sense, the species are more similar to one another in that example. And then the right-hand side is just the opposite case where the mean interaction strength is larger, so competition is strong, but the variation in interaction strength is also larger. And so with this, I can just sample these parameters from probability distributions and simulate this model. I should just briefly say that I choose the pairwise interactions, the higher order interactions, to be scaled inversely to the number of species in an appropriate way. So that when I add up the competition over the whole community, I get something that's order one, so similar to the strength of intra-specific competition. But I'm happy to talk in more detail about how I choose the parameters in the questions. So this is a formal way to construct communities that really don't have any structure or any correlations in their interaction patterns. The only structure is just the average strength of competition and the variation in competition. Okay, so how are we going to determine the effect of these unstructured higher order interactions in this model? First thing we're going to do is just simulate their dynamics until they reach some equilibrium, and it turns out in the parameter combinations I'll mostly focus on, that tends to be the case. They do tend to reach some equilibrium. And of course, we can always just tally up the number of species which achieved a given abundance at the end of a simulation and make histograms like this, which are called species abundance distributions, a very classic way to graph the coexistence outcomes in ecology. And so, you have to, do some high performance computing to get these species abundance distributions. But in addition to just the computational technique, we're also going to use an idea from, or a technique from statistical physics called the cavity method to predict the species abundance distribution analytically using only the statistics of the interactions. And so this is what the cavity method predictions look like when compared to simulation data. You can see they're looking, pretty good. So I'm going to show you mostly predictions from the cavity method as opposed to simulations, hoping that, this slide has convinced you to trust me. So what does the cavity method do? How does it work? It's essentially a generalization of an invasion analysis for diverse communities. So I'll try to give you at least a brief flavor of how this calculation goes in this slide. So you imagine some hypothetical coexisting state where all the species have some set of abundances or the abundances have some statistics. And then you imagine a hypothetical invader to that community. And you just derive what are the statistics of that invader's possible abundances over the disorder in the interactions. And so you can write down some distribution, appeal to the central limit theorem, and you know that it's normal because it's the sum of a bunch of weakly correlated random variables. And then the central sort of guess or idea of the cavity method is to equate the distribution of the successful invaders to the distribution of the coexisting state. So the idea is essentially, if you're a member of the coexisting state, you're like a randomly selected successful invader. And that lets you write down a set of self-consistent integral equations that you can solve in the computer to make predictions for the coexisting state. So there's, of course, some assumptions. You need this community to be diverse. So it's, this theory is really applying in the infinite number of species limit. It helps to have unstructured interactions so that there's not specific correlations between the interactions that will definitely ruin this sort of if they survive in the macroscopic limit, that'll ruin this kind of prediction. And then, of course, you need the central limit theorem to be satisfied. I should say there's some interesting work now pushing this cavity method to incorporate different kinds of structures or correlations between interactions that I'd be happy to talk about in the questions. And so last, just because we are predicting here community assembly as opposed to the stability of some equilibrium point, we're going to use the fraction of coexisting species that survive at the end of the simulation as our metric of coexistence. So before I show you some predictions from the cavity method, I just want to back up for a second and ask, what do we expect for how interactions in general affect species coexistence? And for that, I'm going to consider the simplest possible case in ecology, which is just two interacting species. And I'll tell you some verbal rules which describe the dynamics of this case. So we know that more harmful interspecific interactions give rise to fewer coexisting species. This is just the idea that as the interspecific interactions become stronger, they'll eventually overwhelm the strength of intraspecific regulation and lead to exclusions. We also know that more variable interspecific interactions give rise to fewer coexisting species, and it's for the same basic dynamic. As the interspecific interactions become more different from one another, one species is just a much better competitor than the other and is likely to exclude the weaker competitor. And then last, we know that strong mutualistic interactions can lead to unbounded growth, which we interpret in these models as a lack of coexistence. And so I'll show you these rules remain useful for communities with more species and more complex kinds of interactions. Okay, so we're ready to see some predictions now or some results. So here I'm showing you how the fraction of coexisting species depends on the variation in interaction strengths on the x-axis and the different mean interaction strengths as the different colors and line types for communities with pairwise interactions alone, so as a baseline. And this is what the predictions from the cavity method look like. There's two basic takeaways here. First, that more harmful interspecific interactions give rise to fewer coexisting species. So just that as you go from red to green to blue, you get fewer coexisting species out. And this is just fully consistent with the two species rules I told you about. As interactions become more competitive, you're likely to see fewer coexisting species. And then similarly for the effect of the variability of the interspecific interactions on coexistence, all these curves are decreasing, which means that as the interactions become more variable, some species are more hurt by coexistence, and they're likely to be excluded by the ones that are less hurt. So this is hopefully all kind of intuitive and consistent with the two species picture. And now I'll show you what the predictions for the cavity method look like for higher order interactions alone. And you can see right away that some things are different, but some things are still the same. So once again, all these curves are decreasing, which means that more variable interspecific interactions are giving rise to fewer coexisting species. And I think the basic logic or the basic mechanism for this result is really the same if the interactions are coming from, applying to a two-species system with only pairwise interactions, of course, or to a very diverse system with pairwise and higher order interactions. Variability tends to create winners and losers, and those, just by chance. And the winners obviously exclude the losers. But we found a more subtle effect for the effect of just the average interaction strength of a higher order case. And so specifically, we see that at high variations in interaction strengths, there's a kind of crossing over behavior, where more harmful interspecific interactions actually produce more coexisting species. So this is counterintuitive for how ecologists typically think about competition. So what's the mechanism behind this counterintuitive result? We think the basic idea is that facilitative higher order interactions can actually be particularly destabilizing for coexistence. And so when higher interactions are highly variable but not very competitive, even if they're still competitive on average, we have a large fraction of mutualisms or facilitative interactions that can emerge. And we think that these facilitative interactions create dominant cliques of species which rise to high abundance and then suppress all the others in the community. So we have a few different ways for testing this in the paper, which I encourage you to read. But we wondered if this was a kind of generalization of the third rule that I told you about at the beginning, that strong mutualistic interactions can lead to unbounded growth, and in these models actually reduce coexistence. And so to test this, we simulated a model where the higher order interactions saturated with species densities as opposed to grew indefinitely, so that you couldn't get a sort of quadratic benefit from two neighbors. You could just get, a sort of eventually saturating finite benefit. We found indeed that when we simulated models of this type, we no longer saw the kind of crossing over behavior that we observed for models with an indefinite benefit. Instead, more competitive higher order interactions always reduced coexistence. Okay, so I just want to briefly summarize the answers to this first question. How do higher order interactions affect coexistence? We found that unstructured higher order interactions don't generate widespread coexistence when they're just sort of randomly sampled. It wasn't like we got all the species back coexisting, right? We saw some new behaviors, especially about the effect of the mean interaction strength, and we saw some very similar ones with respect to the variation in interaction strengths. But adding in higher order interactions by themselves don't give you all the species coexistent, which is maybe intuitive. I mean, if they're not going to have any additional structures, maybe that's too difficult of a task for a higher order interaction to complete. Of course, we did find that they could be understood using rules in pairwise theory, especially using this sort of cavity method calculation framework. So on the other hand, we know that higher order interactions can generate coexistence. And we know that from these microbial case studies and their associated theory. So I now turn to the second question where we ask what kinds of higher order interactions stabilize diversity. In other words, in the same generic modeling format, the ones that are more flexible and can be fit to sort of agnostic ecosystems or fit agnostically to different ecosystems. Can we force the higher order interactions to produce coexistence? And then examine the properties of the higher order interactions that successfully do so. So we're just returning to our now favorite ecological model with pairwise and higher order interactions together. And I'm once again going to sample the pairwise interactions from probability distributions with specified means and variances. But now I'm going to force the higher order interactions to produce an equilibrium in this model, because that was why we weren't seeing coexistence in the previous part. These equilibria were, losing feasibility. It turns out that before a given equilibrium where everybody coexisted became unstable as a function of the interactions, they would actually have some negative abundances. And so that's why we were seeing a lack of coexistence in the previous part. So now I'm going to force the higher order interactions to solve that problem. I'm going to force them to create an equilibrium where every species has a positive abundance. And then we can just ask, do higher order interactions that have this minimal kind of structure create an equilibrium that's stable, where there's attraction to the local dynamics? And it turns out that this is still pretty challenging. So if I plot the probability this equilibrium is stable as a function of a sort of metric of the ratio of, or a metric of the strength of the pairwise interactions, this ratio of the strength of interspecific interactions to intraspecific pairwise interactions, it becomes, this equilibrium becomes unstable at a characteristic point. And so, sorry, let me close my window real quick. Apologies. My neighbor is playing music. Oh. All right. The dangers of Zoom talks. Okay, so this system becomes unstable at a kind of characteristic ratio that, of course, is meaningless as an absolute number. But just to give you a sense, the ratio for annual plant systems, where we can actually measure all of the pairwise interactions, is approximately 1. It, of course, depends on exactly how you do the measurements or exactly which species are involved. But this suggests that these kinds of higher order interactions are still not up to the task of stabilizing diversity, right? Just introducing these kinds of equilibrium constraints is not enough to give you stable, diverse coexistence via higher order interactions. And so why is this? I think it's like a fairly intuitive reason. It's just that these constraints really balance the total effect of all the pairwise interactions for each species using these higher order interactions. But of course, stability is really controlled by the pairwise net effects through the Jacobian. And so if we instead chose the higher order interactions to balance these Jacobian or these sort of pairwise level effects, we might see stability. And so that's the next question I want to answer in simulations is just introduce a correlation between pairwise and higher order interactions at this individual pairwise level to see how that affects stability. And so on the x-axis, I'm plotting the strength of that correlation, how correlated is a given pairwise interaction with all the higher order interactions that modify it. And on the y-axis, I'm plotting once again the probability that this diverse system is stable, and then the different colors and shapes are the different variations in pairwise interactions. And so on the left-hand side over here, I think you should think about the first part of the talk where these pairwise and higher order interactions, or I should say these higher order interactions were unstructured. And on the right-hand side, they're very structured. They're very correlated to the underlying pairwise interaction. And this is what the simulation results look like. The big takeaway here is just that in systems that never would have been stable with pairwise interactions and unstructured higher order interactions, we see that structure in the higher order interactions can essentially guarantee diverse, stable coexistence. So that's an interesting result. It suggests that this correlation strength could be one of the key parameters which can actually have higher order interactions produce stable diversity. So to briefly summarize the answer to the second question, what kinds of higher order interactions stabilize diverse coexistence? We found that correlations between the pairwise and higher order interactions can produce stabilizing structures that essentially guarantee stable coexistence. And I think this kind of structure should remind you of some of the examples I set out earlier in the talk, where this kind of antibiotic competition is a clear biological mechanism that could generate these kinds of correlations. So the degrader, necessarily has the opposite sign. The higher interaction that the degrader produces always has the opposite sign of the as the compared to the pairwise competition. But it also doesn't exceed it, right? It doesn't facilitate the sensitive species more than just turning off pairwise competition. And that's the kind of dynamic that we expect to be especially stabilizing. And we've now identified it in this more generic ecological modeling framework. So I think what this suggests or what this necessitates is that we really need experimental measurements of higher order interactions in natural communities to reveal their impact on coexistence, right? Do the experimental systems look more like the unstructured world, where the higher order interactions likely don't contribute to coexistence? Or do they have some specific structures that we can identify and compare to the pairwise interactions and suggest, oh, they're within this other correlated world where they're likely to be good for coexistence? And so to do this, I'm actually going to answer an even more basic question using this kind of classic annual plant system. Specifically, I'm just going to ask, do higher order interactions exist in their dynamics at all? As a sort of first step towards this bigger question of understanding their joint structure with the underlying pairwise interactions. So first, before I can understand their effect on coexistence, I just want to ask, do they exist? And so plants, these kinds of annual plants can compete and intersperse in diverse environments where there's a large potential for higher order interactions. So here I'm showing you, this cute picture of all these little plants, and each of the different colored toothpicks is a different species. So these plants will experience competition from a broad range of neighbors in their lifetime. And past work has leveraged the kind of discrete generations in the system to make a lot of progress measuring pairwise interactions. So a sort of previous experimental workflow will be to grow these annual plants in different plots with increasing numbers of background species, or I should say background individuals of a given species. So on the left-hand side of this diagram, for example, a focal teal plant is being grown by itself. So you can directly estimate the sort of discrete analog of this RI parameter I've been telling you about, just how many seeds you produce in isolation. And then there's some increasing density gradient of a given background species, this red species. And then by counting up the number of seeds that these focal plants produce at the end of the season, you have a direct estimate of the fitness of that plant, which is actually a very unique measurement in biology. It's usually pretty difficult to get the exact fitness of a given individual in a system. And so this is a nice system for doing that. And then you can just fit some curve to the decline in fecundity with the increasing density of background competitors to get these kinds of interactions, these pairwise interactions I've been telling you about. So there's a little diagram of, what that might look like. And so this sort of slope of the decline in this curve is a proxy or is a, equivalent to this kind of Aij or alpha ij parameter. So it's very difficult to do these kinds of experiments for higher order interactions. I mean, it's possible, but it's challenging. I mean, and one reason it's challenging is because there's some model inference issues, right? So in the pair, we don't really even know the right models to describe the pairwise interactions. And if you get the models for the pairwise interactions slightly wrong, you might infer higher order interactions that are really due to some poor model fit issue, as opposed to a true biological higher order interaction. I think there's some ways around this. But it's still, a pretty challenging problem. Maybe even more fundamentally, there's a big problem, which is just that it's a huge amount of work to measure the pairwise interactions in this way between, say, like 5, 10 species of plants. And so it's combinatorially worse to study higher order interactions, of course. And that motivated me to try to design a model-free test of just whether higher order interactions exist amongst these annual plants, as opposed to measuring their exact interaction strength. And to do this, I wanted to use the limited spatial range of competition between these plants to try to manipulate or toggle up and down the frequency of pairwise and higher order, or sorry, I should say of higher order interactions. So the idea is that here's some focal plant that's competing with this interspersed neighborhood of green and purple background competitors, and each of these background competitors, as well as the focal plant, has some characteristic radius around which they experience competition, which we can just measure in the field at something like 10 or 15 centimeters. And so in this case, it looks like the kind of classic higher order interaction situation that I told you about at the beginning. This green individual is competing with a purple individual in the background, as well as some members of its own species. And because it's competing with this purple individual, its phenotype might change in some way. Say it roots more deeply or it grows taller. And this can have some cascading impact on the focal plant. It could change its competition relative to a case where it wasn't competing. It can make it stronger, say. And so our idea is to grow these exact same plants in an alternative spatial arrangement, which toggles down the frequency of these interspecific higher order interactions. And so this was the basic experiment we did, is we grew the same exact identity and number of plants in these two different spatial arrangements. So on the left-hand side, these green individuals, because of the limited spatial range of competition, never compete with members of the other species, or at least only do so at the border. Whereas on the right-hand side, those kinds of interactions are often happening. And so the idea is just that there's many fewer interspecific higher order interactions on the left as compared to the right. Importantly, in these two spatial arrangements, the focal plant is seeing the same competitive environment. They're seeing the same number of purple or green individuals, whether they're clustered or aggregated, like on the left-hand side, or if they're intermixed together, like on the right-hand side. So we've preserved the pairwise effect of the background competitors on the focal plant. And that means that we can just directly compare the fecundity of focal plants between these two spatial arrangements to reveal the strength of these kinds of spatially mediated higher order interactions. So what are the different possible outcomes of this comparison? Well, the first thing is that we could see that the focal plants produce basically the same number of seeds between the two spatial arrangements, which means that we would have no evidence for higher order interaction in the system. And I should say, before we ran these experiments, or maybe more accurately, as we were running these experiments, we talked to a fair number of plant ecologists, and this is what they predicted. So this is definitely a real possibility that this spatial arrangement would have no impact. Alternatively, we could see that the focal plants produce more seeds in clustered relative to mixed environments. And this suggests that higher order interactions exist because there's some difference between the spatial arrangements, but also that they're competitive because when they're operating in the mixed treatments, the focal plants produce fewer seeds. And then, of course, there's the alternative, which is that they produce more seeds in the mixed relative to the clustered environments, which suggests that higher interactions exist and are facilitative. Okay, so to actually, we didn't just do these experiments with dots. We also did them with actual annual plants at Sedgwick Reserve outside of Santa Barbara. And I want to shout out Zach Cold, my co-first author on this project, and who's really instrumental in its success. So this is what the annual plants look like in real life and not in dot form. So on the left-hand side, hopefully you can tell those are a bunch of clustered annual plants. And then there's focal plants along the center line of that plot, along the center line of the graph. So you can see in the sort of diagram format, but then also in real life, we have a bunch of acmaspon on the left-hand side clustered, a bunch of Europapus on the right-hand side that's clustered. And if you really look closely, you can see a salvia bulb right there in the middle of the plot, which is our second focal species. Hopefully you can see my cursor. That would make this easier to see. On the right-hand side, of course, we just have these two same species randomly intermixed together, and you can see you can barely even see the acmaspon in this plot because the Europapus is much taller. And so we used 5 different background competitors to produce 10 different background species combinations. And we, importantly, used an equal number and the same identity of competitors between the treatments. Really only the spatial arrangement differed. And we went back in and counted up the number of competitors the focal plants were competing with between the two treatments, and then removed plants to remove any systematic differences between the two treatments and the number of background competitors. Okay, so now we're ready to see what the effect of this spatial aggregation was on the strength of competition. So on the x-axis here, I'm plotting the different background species combinations. So, which of the two species were in the background. And then on the y-axis is just a big Bayesian linear model, which reveals the effect of aggregation on focal species seed production. So if you're above this 0 line, it means that focal plants produce more seeds in clustered relative to mixed environments. Of course, if you're at the 0 line, there was no effect, and then if you're beneath, it's the opposite. Good. And then I sort of took 3 different focal species, but aggregated them together in this statistical framework. So that'll just show you results for background species combinations. So this is what the effect of the first background species combination looks like. It turns out that when acmaspon and festuca are the background species competitors, focal plants produce more seeds when these 2 background species are clustered relative to mixed. So this is exciting, right? This suggests that these species are producing a higher order interaction. If you spent your whole PhD studying higher order interactions, it's good to see that they at least sometimes exist. amongst these plants. And it turns out that this is actually the typical outcome across these background species combinations. So I've colored all of these posterior densities orange when they're above zero, and that's the most common outcome. So that there are higher order interactions amongst these plants, and they tend to be of the competitive flavor, of the competitive variety. So across all these different background species combinations, it tends to be the case that the focal plants produce more seeds when their background competitors are clustered relative to when they're mixed. Now this result, so yeah, they're competitive. And this result depended on the exact identity of the background species, right? So whenever Acmosmon was one of the background competitors, we saw a strong higher order effect. And the same was the case for when Salvia and Europapus were the two background species. By contrast, when Festuca and Plantago were the background species, the focal plants didn't really care in terms of their fecundity whether those competitors were clustered or were mixed together. And we'd like to know, why is this? What explains the variation in higher order interaction strength across the different background species competitor combinations? And to try to understand this, I'm now going to, make more practical this kind of diagram, or this sort of toy picture I've been showing you throughout, where it could be the case that when the higher order interactions are operating, either one of the background competitor phenotypes might change, right? So they might root more deeply in the soil because they're competing with another species, or grow taller to avoid competition from light from this other species. Um, and now we can just directly measure all of these plants in the background's phenotype, or their height or their biomass, similar sorts of metrics, and compare them to the other spatial arrangement where they were only competing with members of their own species. So that's the idea. We measured a suite of what are called functional traits in the plant ecology literature, like the specific leaf area or the canopy index, which, as an example, is a metric of how tall up versus how tall out these plants grow. And then we also measured some performance metrics like their height and biomass. Then we put all of these functional traits and performance measures into PCA so we can kind of quantify the overall phenotype of a given Acmosmon individual, say, in the background. And then just by measuring how much along, say, the first PCA axis these phenotypes moved when comparing the cluster to the mixed environments, we come up with an overall metric for how phenotypes shifted as a result of spatial aggregation, right? So that's the sort of big picture, is that we now have a way to decide how much did the background competitor traits change as a function of the spatial arrangement. And then we can just ask, can we predict the strength of higher order interactions that I've been showing you previously from these changes in background competitor phenotype? And indeed we can. So this is showing the relationship on the x-axis between the difference in competitor size and traits that I was just telling you about. And the y-axis is just the effect of the higher order interactions that I was showing you in the previous slides. And so we can see, at least by ecology standards, this is a pretty good correlation between these two metrics, suggesting that as the background competitors become more different from what they would have been in a purely intraspecific or a purely single species system, they also produce larger higher order effects. And so it suggests a possible mechanistic underpinning for how these higher order interactions might come about. They probably come about from some phenotypic shifts in response to competition. Okay, so I just want to briefly summarize the answer to this third question. Do higher order interactions structure the dynamics of annual plant communities? We found that the neighbors of the neighbors modify plant fecundity, suggesting that there are kind of competitive higher order interactions in the system. We also found that phenotypic responses to competition can predict higher order interaction strength. So it suggests a possible mechanistic underpinning for these higher order interactions. I think the big picture takeaway here is that higher order interactions of this flavor might be more common than we previously thought. And that's because aggregation and the lack thereof are very common in plant communities, right? There's all kinds of different other ecological processes which might create aggregation or then erode it depending on the circumstances. And similarly, phenotypic plasticity is also incredibly common. So if the combination of these two processes can modify competition strength, that sort of dynamic might be playing out in a lot of natural plant communities worldwide. So now I just want to briefly summarize the takeaways from my whole talk. We first found that unstructured higher order interactions likely don't generate widespread coexistence, but could be understood using some classic pairwise theory, specifically motivated by the cavity method. And then we found that specific correlations between pairwise and higher order interactions can stabilize diverse coexistence. And then last, we found that higher order interactions are pervasive amongst annual plants. So what progress have we made on this bigger picture question I set out to answer, whether higher order interactions explain coexistence in diverse ecological communities? Well, our theory suggests that they can, but only if they have specific correlation structures. And then our experiments show that they existed, but it's very difficult to test coexistence in these systems of annual plants. And that's because, although in 1 year I can get pretty good estimates of how strongly the plants are competing, it takes something like 50 years to actually measure coexistence, to measure the assembly of, a given set of species experimentally. So just to briefly say, I think the big, next step here is to come up with fully parameterized models of pairwise and higher order interactions derived from high throughput experiments. And that's what I'm trying to do now in my postdoctoral work by measuring the interactions between microbial strains which compete and coexist in the plant phyllosphere, so bacteria that live on the surface of plants. And the nice thing here is that we can use this automated pipetting system to infect many plants very rapidly. So this is, this little pipette tip here is picking up different bacterial combinations from the wells at the top of the video, and then just dropping them down onto a bunch of plants which are grown in these well plates. And so with this system, we can infect something like 10,000 different plants with defined microbial communities in 1 week. So that's something like 10,000 different ecological communities that we can then study. And then we can sequence, so all these plants and bacteria have barcodes inserted into their genomes, which means that this kind of sequencing strategy lets us estimate community composition very rapidly. And so just to give you a flavor, 1 week of infection time for these bacteria is equivalent to about 35 years for annual plants because, they're much faster generation times. So I'm very hopeful that this kind of experimental system will let us measure pairwise and higher order interactions together and also validate these models predictably while measuring coexistence. And then we can really just directly account for, to what degree are these things structured and to what degree do higher order interactions account for any coexistence we observe. Okay, so with that, I just want to thank everybody for listening. Thank you, thank my collaborators, and thank all of my funding and institutions. So thanks for listening. I'm happy to take any questions.
Thank you very much. Very, very interesting talk. I'm really looking forward to see the results of your current experiments.
Me too.
So, yeah, I can imagine. So yeah, if anybody has questions, please unmute yourself and go ahead. And don't
So
be shy.
maybe I can start with a question, if you
Sure.
don't mind. So thank you for this interesting talk. So I have, in fact, two questions and one comment, if you allow me. So first of all, you've mentioned that the correlation structure can influence, the existence of correlation. Will make this coexistence happening. And I was wondering, have you considered the relation at the trophic level in ecosystem? So whether there's some correlation.
Yeah, that's a great question. So I personally have not worked on the trophic structure, but it's actually the situation where I think the most work has been done in the higher order case. So there's these very nice experiments where, well, nice is the wrong word. There's these kind of mean experiments where they basically have spiders predating two different kinds of mites, and they'll like sew the spiders' mouths shut, and so they can compare the case where the spiders can actually eat the prey versus where they can influence their behavior but don't consume any prey. And so that's very clear evidence for higher order interactions. Those are usually called trophic. They're called either trait-mediated interactions or trophic interaction modifications. And so there's actually a big body of literature studying those kinds of behavioral dynamics that are induced by, say, predation. So that's one big area. I personally haven't studied those things, but I think you're completely right that those will naturally have correlations, right? Where there'll be some, maybe a very correlated block with another very correlated block, that kind of thing.
Yeah, I was wondering whether your results will be just correlated with the trophic level in some sense, because I was wondering whether you're taking just the random network or structure in a way that it's similar to the trophic network, because I mean, there are some rules in some sense, right? Yeah, thank
Yeah.
you.
No,
Uh,
yeah, totally.
yeah, yeah.
So maybe just, yeah, go ahead.
Yeah, so the second question is that you've mentioned that these higher order interactions are more popular than we initially thought, and I was wondering what is the lesson for us. So, do you think that if we measure the relation between the species, do we measure the final effect of these higher order interactions, or do you think? I mean, so do you think that inclusion of this higher order interaction in our consideration will make the significant difference in our way how we think about how the ecosystem behaves in some sense? Yeah.
Yeah, it's a great question that I don't completely know the answer to. I would say I think they're more common than so in this sort of standard logic that I was describing at the beginning, a lot of these, especially plant ecologists studying coexistence will isolate and do all pairwise experiments, and therefore they'll never measure the strength of the higher order interactions. But there's a whole other literature fitting these kinds of models to existing observational communities where you can't, a given focal plane is competing with a very diverse milieu. And I think those experiments or those studies sort of naturally are also fitting to the effect of higher order interactions. So I kind of think those are two different empirical settings where it's like both using pairwise models, one which you only ever have pairwise information, and one where you have pairwise and higher order information, but forced into a pairwise framework. And I do think that there's some work to be done comparing those approaches and asking, are they, do you see systematically different pairwise interactions in this world where they're also considering the higher order interactions, but sort of inadvertently? I think of the big picture, so as a kind of like maybe a downer result about the higher interactions that we identified in the experiment, I think they will modify things like biomass and the abundance of the species. I don't think they're going to resolve coexistence in that system. My guess is that coexistence in the annual plant system is due to spatial heterogeneity across the landscape. But that's sort of a different experimental approach. In the microbial settings, I think it's a little bit more up for grabs. Their higher order interactions might be playing a really crucial role for stabilizing coexistence, but that's the kind of thing that I want to work out exactly. Yeah.
Thanks. And just one last thing, I just wanted to share the information that together with my working group, we are working on the notion of pangraph, which is exactly what you can see on your picture. So there is a kind
Yeah.
of theory. Oh, you know it, maybe.
No, please go ahead. I don't know it.
Yeah, so mathematical framework for the networks is such that there might be the vertex that influence the edge, and also there might be the framework that one edge influence another edge, and we're studying centralities. So I just thought it might be of use to you. Thanks. Thanks for
Oh
this
yeah,
talk.
thank you. I appreciate that. I'll check it out. Yes, thank you. Thank you for your questions.
Yeah, great. Other questions?
Maybe I can go ahead.
Yeah, sure.
I have just a couple of curiosities. So in the beginning of the talk, so first of all, very interesting work, very cool work to be honest. So you were mentioning that when you are trying to find coexistence in the pairwise case, and the reasons why you don't find that is because of interaction chain, and higher order interactions. So the first thing, so the confusion is the thing with the interaction chain, which is basically pairwise interactions, if I'm not wrong. So why doesn't the previous model on pairwise interaction exhibit interaction chain? Why is interaction chain not seen in pairwise models?
Oh, okay, yes. So I should say it definitely can be seen in pairwise models, the interaction chain, but
Okay.
it's an example of a very structured correlation, right? So the sort of rock, paper, scissors dynamic that would never emerge, or I should say, that will only emerge in these random models if you build in a correlation structure, right? So you can
Okay.
have the kind of generalization, the messier version of the rock, paper, scissors game in these models that I was talking about, but only if you
Yeah.
like introduce an explicit correlation. Yeah.
No, okay. Yeah, perfect. And the other thing was when you use the terminology unstructured, do you mean that we don't know the exact dynamics of the higher order interaction, or is it something else? I just want to get a better clarity of the word unstructured when you mentioned that.
Yeah, so yeah, it's always a bit, I think I do a bad job. Well, okay.
No, it was just me being dumb.
It's always a bit challenging to like describe exactly what I mean by. So in these models, I'm basically completely randomly sampling all the interactions from a distribution with a
Okay.
mean, right? So everyone, if there's no variance at all, everybody has some intra-specific, within the species competition, and then some
Yeah.
interspecific competition strength that's lower. And
Okay.
then I just introduce some variation around that interspecific strength. So it's essentially a completely random network, right?
Okay.
And then you get, patterns at the level of species abundances and who coexists from that. So that's kind of what I mean by unstructured. And that I should say is like a very coarse picture of what ecology should look like, right? Like no one really believes that's what the species interactions are. The question is, does that picture still provide some predictions that can be tested, or gives us some like nice null model for especially how the very diverse communities behave? And so what I mean by structure is then adding in some kind of correlations to that picture, where say, a kind of classic structure in ecology is a competition colonization trade-off, where you might be a very good grower, meaning that your, RI, your growth and isolation term is very large, but you also suffer a lot of competition, right? And so that would introduce
Okay.
a correlation both between the interactions you experience, but also the interactions you produce amongst the other species, right? So that gives you a pretty structured kind of A matrix where there's like, some of the values in the first row are very large, and then the first column is very small, et cetera. So something like that. And those dynamics definitely occur in ecological communities. The question is just how prevalent are they? How strong are they? And so we have, a lot of ecology, I would say, is guided by these kinds of case studies where you like found a given community that did that. And so we know that happens. But if you go to a randomly chosen community and measure the interaction patterns, it's less clear how competition colonization it'll be, or if it was going to look much more unstructured. So in general, in these annual plant communities, they look less structured than probably you would have expected. And that might be to do with how we do the experiments. Um, so
Okay.
that's a potential limitation, but yeah. Anyway, so maybe a
Yeah,
very
yeah.
long answer about, yeah, okay.
No, I get the gist of it. So yeah, perfect.
Okay.
And the third one was when you say that AIA is equal to 1, you mean that you're talking about intraspecific interactions, if I'm not wrong. Does BIII, which is basically does that have any physical significance? Like, does that have a physical interpretation? When you
Yeah,
say
that's a great
B,
question.
okay, so
Yeah, so we set all the BIII terms to be 0. And that's
okay,
because we're sort of assuming that the underlying pairwise logic can describe intraspecific dynamic, right? So when you do these kinds of
yeah,
experiments, the first thing you do is do sort of monoculture growth curves. This is in the microbial setting. And if you need like triplet terms to describe that, something is a little weird, right? Because that doesn't have a clear, yeah, as you say, biological, physical interpretation as like, I'm modifying the effect of someone else on me, but everyone's the same. It's a little bit weird.
yeah,
So in the logic of the theory, we just ignore those things, but we also test their effect, and they have a strong impact. If you have self-regulation that's cubic as opposed to intrust, as opposed to quadratic, then you don't get these same facilitative impacts, right? And it's because the strength of the higher order terms can now be of the sort of facilitative higher order terms can be compensated by cubic self-regulation. So that it's definitely a key
mm,
parameter. Yeah.
okay. And since we are on the topic of higher order structure, uh, have you ever looked into higher order motifs? Like when you, in the case of graphs, we have motif structures which are quite relevant in ecology, like structured patterns in networks, which are like quite have a high probability than a random network. So have
Wait,
you ever
sorry, can you
looked
say it
in,
again?
motifs in graphs, in directed graphs, which are like
Mm-hmm.
patterns which occur more with a more probability when compared to a random network, something like that. So have you ever looked into the direction of higher order motives? I don't know if such a thing exists in the literature, but I'm just curious in this particular case.
Yeah, it's a great question. I know of some recent papers that incorporated some of these trophic interaction modifications, um, like amongst 3 or 4 species, and said here are some like basic motifs that could emerge higher order wise, and like assign them biological meanings, and then simulated stability in those systems. But I think that's a great open question. I mean, I think it'd be especially exciting to try to identify higher order motifs in real empirically measured interactions, which is still, kind of unfortunately like where we are. We don't have very many of those.
Okay.
So that's probably the first step. But like, I think that's a very interesting question because if there were some, even in the pairwise world, like if you found rock, paper, scissors to be more common than you would expect by chance, that's sort of indicative. And the basic result there is that they don't seem to be super prevalent rock, paper, scissors dynamics. But if there were some higher order ones that were, that'd be really exciting. But so I guess the answer is I don't know. But I know there's some
Yeah.
theory about the motifs.
Yeah.
Yeah, yeah.
Thank you.
Thanks.
Okay, any other questions? I see there's one in the Q&A. I don't know if Christina,
Okay.
if you want to unmute and ask it directly.
Yes.
Okay.
Hi, everyone. Great presentation. I was just wondering about the just plant interactions in general, like how they communicate and how you measure that, like their hormones, like do they communicate through the air, through the roots? And then also, do you look at root biomass? Because you said you're looking at biomass, but I know it's hard to like pull them apart, right? And quantify that. And then if you're if you're comparing plants grown in soil versus plants grown in on a plate, they will have obviously different metabolic output measurements, right? So I know, yeah, that's harder to quantify, but those are ways that they communicate and influence each other, right? So then I guess measuring root exudates might be a way around it. And then like similarly, are you looking at the soil microbes at all? Or considering their influence? And that's more like I'm thinking mycorrhizal networks, but that's more relevant for forests or, longer than annuals. But yeah, thanks.
Yeah, thank you. That's a great question. Yeah, so in terms of the practical stuff, we, as you suggested, only measured above ground biomass because it was such a mess to get the roots out and like be confident that the roots were actually from the plant that we wanted. That's something that we really wanted to do. We really wanted to try to take out a given plant and or to look at its roots and also to see if the roots had some spatial structure, like if they were growing away from the dominant competitors, right? Because I think that would have been a very clean mechanism potentially for like, oh, you can just see that like when the plants are all clustered, say, let me see, like this Aquaspan guy is really competing only with itself because it doesn't like this dominant Europapus competitor. Um, so I think the big answer is basically we would love to do some of those root comparisons. We'd also love to do some of the mycorrhizal soil microbiome comparisons, but we just didn't have the bandwidth to do those in this experiment. One thing that we did find was that this Aquaspan in particular um, has a plastic growth morphology where it essentially it can grow more out or more up. And when it's growing in the mixed treatments, it tries really hard to grow up, but it basically, and it does way worse. Like it's just suffering way more from competition. Whereas when it's competing with itself, it's growing really far out. So that's like, and then it does better. And we think that's like one of the big mechanisms for these higher order interactions, these kind of growth form plasticity morphology, like, these sort of plastic responses at the level of morphology. And we think those are almost certainly happening below ground and or they could be microbially mediated as well, but we just didn't have the bandwidth to do those things. And then just for your last question about um, sort of growing them in media versus growing them in soil, I completely agree. They would definitely it would be very different. When I'm growing them in media, I'm mostly studying the bacteria as opposed to the plants. So that like gives me a little bit of hope that like at least as far as microbial experiments go, this is much more natural than like glucose in a test tube. At least there is a plant there, but it's
Yeah.
still not the full picture. Yeah.
hmm hmm Um, and do you think that maybe so if someone were to measure root exudates in an experiment like yours, would let's say certain soil probes that you keep inserted there that take measurements periodically, or just I'm trying to think how one would go about measuring those.
Yeah. It's a great question that I'm not a real expert on. I think you're right that there are soil probes, and then you can do some chemical analysis of the soil. You can also, of course, sequence the soil. You can take soil samples and then, purify, extract, the bacterial DNA and sequence. The big approach there oftentimes, so before the kind of effect of soil, um, I guess I just say like the composition of the soil being curated by the plants has been studied for a long time, and it was it was even we were even doing it like before we were really particularly good at sequencing. And there, the approach was often you have some sterile soil, you also have some soil where you grew a given species, and then you remove the plants, you took the soil, and then grew another species in that same soil and compared it to some reference soil. And so that gives you some idea at the species level. You don't know what actually happened in the soil, but you could say, okay, species one makes the soil bad for species two, or vice versa, and then you can make some predictions on that basis. And so that's definitely an ongoing body of work too.
Very cool. Thanks.
Thank you for your questions.
Okay, any other questions from the audience? Okay, if not, I have one if you still have a bit of time, or I don't know if you're
Please.
busy. Okay, now mine is just modeling questions that is kind of related to the thing that we're mentioning. So the fact that there is a soil, there are external factors and so on. Wouldn't it be useful or reasonable to include some let's say external field term in the dynamic equation, be it Lotka-Morterra or whatever else, like something that couples the species with each other through a shared environment. So,
Yes, definitely. Yes. I think that would be a good thing to do and is a good future step. I think it's actually particularly interesting in the higher order case because there's these sort of verbal arguments that communities structured by higher order interactions are kind of more susceptible to perturbations because they can propagate through very quickly or something like this. And there's one very nice recent preprint that studies a related question, but I think in general the question or the yeah. So we have those kinds of results theoretically for the pairwise world.
okay,
Not even as much as you would think for the diverse world. Like in general, it's harder to study these sort of end species systems. So I actually don't know too much about the current status, but it's almost always the case that if you add in a higher order interaction, you could always just do the equivalent, uh, you know,
okay, yeah,
ecology theoretical modeling project and see what happens. And I actually think that's a situation where the higher interactions might be pretty different than the pairwise interaction. So basically, I think it's a good idea
okay,
that I
okay.
haven't done it for simplicity. Yeah.
Makes sense. Thank you.
Thank you.
Okay, then, if there are no more questions, I'll just stop the recording.